Okay, so in the last video we were done till the only temperature, like we are discussing factors that affect activity of enzymes. So we are done till temperature. In today's video we are going to look into pH. So as I just told you, they are very sensitive. They need very optimum temperature pH to work properly. Okay, so if I just provide them like this middle point is the optimum temperature. This one part would be like where they get inactive, and the part where they lose everything, that is denaturation. And pH, like enzyme need very proper pH to work properly. And if they just get change in pH, this means they just get change in their activity. Like if they just require two, like activity 2 pH, and they just got 5, so this means they are denaturized and they need a proper specific to work. So we are having different pH for their work, and that pH change could be temporary or permanent as well. So if we just take example of protein, which are made up of amino acid, then these amino acid are like ionized, or they just get denaturation. This means like they are very if I'm just taking example of protein, which are made up of amino acid. So if they just required a very low temperature and they got very high pH, not temperature but pH, they got very high, so they will get ionized, like ionization will be there. This means if there's ionization, so they will lose their power, that will be denaturized. And denaturation is just right here. So I've just written here example, the pepsin, which is secreted in the stomach. Oh my god, which is having pH 1.5. This means that is acidic. So like here we have the pH scale. The middle point is 7, which is neutral. Here we have the 1, and the last point is 14. So the 14, like above than 7 to 14 are basic, and 1 to 7 are acidic. Do not understand A or B. I'm just writing it's acidic and this one is basic. So this was the example. Then we also have few other, like this is enzyme concentration and the substrate concentration. Okay, so they are very sensitive, as I just told. So if you just need two substrate and there are the two enzymes, so this means this is done. But if you just provide them more, this means the enzyme concentration is supposed to change itself. How? If I just make a graph, okay, the activity which we can see is like this, okay, and till this the downward parts just tells concentration of substrate, and the upper one part just show the like this show the maximum activity till the upper part. Okay, but what if you just need the activity till this one part? Let me just change color. If they just need this much activity to be done, so what will then? So we will divide maximum into 2, like maximum would be divided into 2, so we can just get the concentration. So this means they need a particular concentration, as we done competitive and non-competitive inhibitors is all. So they were also like telling, or which the one whose concentration is more, they were supposed to add, like they were about to add. Okay, so the same goes here. They need to have a particular enzyme concentration to work properly, and the activity depends on the substrate and enzyme, as I just told you. And if the substrate is high, so that will attach directly proportion to the substrate of enzyme, and these all. So now the last one which we just have is the substrate concentration. In this one we were just discussing the enzyme concentration. Here we are discussing substrate concentration. So substrate concentration, the rate of reaction increase with an increase in concentration of substrate in a condition where enzyme concentration remain constant. So if you just graph, and this is the complete optimum temperature they require. But what if we have provided more? So on this one stage, like this one stage, they got denaturization. This means you can read this as well. Increasing concentration does not affect the reaction. Then if you just increase concentration, this will not really affect the activity of enzyme, just because they have gotten the optimum temperature, rate of reaction they were supposed